welcome back, everyone. I'm here at school 1 week before you have to come back to help get you prepared both for your upcoming unit test on heredity and also the upcoming your upcoming AP exam. This is intended to be a review video. It's not intended to go through and define for you all the keywords for the topic of meiosis. So I'm expecting that you have already checked out those awesome videos on College Board where they define for you clearly haploid, diploid, gamete, what is meant by the letter N in our notations on meiosis, crossing over, independent assortment, and fertilization. If you don't know what those keywords are, pause this video, go review those, and make sure you have a deep understanding about them before you check out this video. Okay, hopefully everyone is on the same page now, and we're going to review this topic and make sure you're fully prepared for both your unit test and for the AP test. Okay, so since we already understand that meiosis is not just 2 rounds of mitosis, that's kind of the first thing to understand. It's similar. It uses a lot of the same cell machinery. However, it's not just 2 rounds of mitosis, but it looks like that. That's kind of a level 1 understanding of meiosis. It is basically 2 rounds of mitosis. Well, once we really get into it, we realize that it's different in some important ways from 2 rounds of mitosis, in that because we're ending up with 4 gamete cells instead of 2 identical cells, we have some important differences that lead to this genetic diversity. And those differences are what make meiosis unique, because even though we're using the same processes, prophase, metaphase, anaphase, and telophase for mitosis, we are not doing them in the same way. Again, let me repeat that again. Prophase 1 is not the same as prophase. Prophase 2 is not the same as prophase. And prophase 1 is not the same as prophase 2. They are all using the same cell apparatus. They're all using the spindle. They're all doing the same thing, doing a similar thing, and that's why they all have the same name. They're all condensing chromosomes, but they are not the same at all. They all are different processes used for different things. And one of the key differences, what we're going to talk about, is crossing over occurs during prophase 1, which does not occur in prophase 2 and does not occur in prophase and mitosis. So we see that even though these things are similar, they all involve the condensing of chromosomes, they're all different. They're all different. And so we need to understand and what I would call level 2 understanding of meiosis is that it's like mitosis, but it creates 4 gamete cells that have half the genetic information, which we call haploids, as opposed to mitosis, which creates 2 identical diploid cells. Let's look at what that looks like in your handout you're going to be studying here. So we see in this picture that in meiosis, we start prophase 1 very similar to prophase. The nuclear envelope starts to dissolve and the chromosome form. So far, we're actually looking identical to prophase. However, a key difference here is that in prophase 1, unlike prophase and mitosis, they actually form something called a tetrad. And this is where crossing over occurs. The important thing is only homologous chromosomes come together. This is a carefully regulated process that ensures that crossing over does not lead to the deletion of important genetic information. You can't just randomly exchange chromosomes. You need to only exchange chromosomes that code for the same information. To oversimplify, which you've learned in the other videos, this is not actually the case, but let's say there's a gene that controls how tall you are. Again, that's not really the biology, or oversimplifying. If there's a gene that's a tall gene and a short gene, those 2 genes could get switched. We don't want to switch the gene that controls height to the green with the gene that controls how red blood cells are made. Otherwise, some gametes would not have that important information. Some would have 2 genes for red blood cells, and some would have 2 height genes. That's not useful. We need to exchange similar genes in our crossing over. That occurs in prophase 1. And then the rest of the process is pretty similar to mitosis in some ways, but a major difference is, again, crossing over has occurred. And so we end up, instead of with 2 identical cells, we end up with cells that are both we end up with haploid cells at the end of meiosis 1, but they are not identical, and crossing over has occurred. And there's a few questions about that. So I would definitely study the video on College Board, study this handout, make sure you really understand meiosis 1. In both meiosis 1 and meiosis 2, then we get to the important idea of independent assortment. And in independent assortment, we see that in gamete formation, the chromosomes can come from any origin, meaning, and it's nicely color-coded for us here, meaning that each chromosome has 2 chromatids. And when the chromosomes are separated in the anaphase, either half can end up in any of the gametes. And we're going to see that in a cool classroom demonstration when you come back next week. So hopefully you have a better understanding of independent assortment, but independent assortment has to do with which chromosomes end up in each gamete. Remember, humans have many chromosomes, and so, even like a fruit fly has many chromosomes, but humans having 23 chromosomes, one of the key features of diversity is that if every time the left-sided chromosomes always ended up together and the right-sided chromosomes always ended up together, they weren't independently assorted, so lefts always went together and right always went together, then that would be much less diverse. But rather, we're going to get a random numbers of lefts and rights. And when we have so many combinations possible, which would be 2 to the 23 possible combinations to do this, we're going to get a great amount of diversity. That's called independent assortment. Where each sister chromatid goes is independent of the other ones. And lastly, in fertilization, which again, we're going to focus on humans for a second, because the details of meiosis in other animals and in plants are a little bit beyond the scope of this class. Fertilization is the last step that leads to genetic diversity, and we'll talk about this in detail in class. For the AP exam, again, you just want to remember and really understand what is crossing over, what is independent assortment, and what is fertilization, how these lead to genetic diversity. This one's pretty easy. Through crossing over and independent assortment, we got a lot of diversity of gametes, meaning that no 2 sperm cells or no 2 ovary egg cells in humans are the same, even in the same person. So each one is unique. And so when you have the combination of all those possible sperm and eggs, you end up with great diversity, which is why that I'm very different than in my siblings, even though I have 6 of them. No 2 of us are the same. That's because we're all come from a unique sperm and egg, unless you're identical twins. Okay, so what would be, to wrap up this, I want to look at some quiz that I gave my students just for a quick review. So at each point, after each of these questions, you can pause the video and try them for yourself. So question 1, where does this occur? A cell divides to produce 2 daughter cells that are genetically identical. Keyword is genetically identical. Hopefully you said A, this is true for mitosis only. Meiosis does not create genetically identical cells. Homologous chromosome synapse and crossing over occurs. This occurs only in meiosis 1. Crossing over occurs specifically in prophase 1. Second, when does independent assortment of chromosomes occur? Well, the chromosomes actually separate out in mitosis 2. What is a genome? Well, a genome is the complete, all of the genes in an organism. Which of the following statements about genes are incorrect? Is genes are segments of DNA. They contain information for synthesis of enzymes and other proteins. But there are many genes on each chromosome. And lastly, let's look at 8. If the liver cell of an animal has 24 chromosomes, how many chromosomes does a sperm have? The important thing here is that through independent assortment, the sperm and the egg are both have n number of chromosomes. In this case, the animal's 2 in number is 24, so the n number is 12. And so I wanted to point out some of these review questions for you. I would say these are really, the purpose of these questions are really just making sure you understand the keywords. On the AP exam and on your unit test, I'm going to ask you to go a little bit deeper than those type of questions where you really understand what is meiosis. Okay, so to wrap up, I'm going to give you what I would call a level 3 understanding of meiosis. Again, level 1 was that meiosis is kind of like mitosis 2 times. That's a really basic understanding and not going to get you very far. Level 2, we added the fact that the product of meiosis, unlike mitosis, is 4 genetically distinct gametes. That's good. That's understanding the key difference. However, level 3 would go deeper. And this is how I would say, this is what I would say is a level 3 understanding of meiosis and mitosis. So in meiosis, we have prophase 1, metaphase 1, anaphase 1, and telophase 1. In prophase 1, the chromosomes condense and crossing over occurs, which makes it unique from mitosis. In crossing over, homologous chromosomes are exchanged, and this creates the majority of genetic diversity that results in mitosis in prophase 1. Then those exchange chromosomes are pulled apart and separated ultimately into the 2 products, the 2 cell products of meiosis 1, which are genetically distinct 2 cells with still the same number of chromosomes. Okay, after that, in meiosis 2, those 2 new cells immediately go through another round of prophase, metaphase, anaphase, and telophase. We call that prophase 2, metaphase 2, anaphase 2, and telophase 2. In this case, the 2 sister chromosomes are actually split apart, and this is unique because in telophase 2, we actually end up with 4 haploid cells, which are independently assorted, in which each chromosome was independently assorted from each other. And so we end up with a great possibility of millions and millions, more than millions, but millions and millions of possible egg and sperm cells because of crossing over and independent assortment that occurs in prophase 2, metaphase 2, anaphase 2, and telophase 2. That's what I would call a level 3 understanding of meiosis. I encourage you to put in the chat, what is your understanding of meiosis, and would you consider it a level 1, level 2, or level 3 understanding? Or if you're a biology expert, maybe you can give us a level 4 understanding and help me to understand it better. Okay, that's it for today. I hope that's a helpful review. Again, the purpose of this video is not to define all the terms for you. I assume that you're already doing that on your own, but to give a deeper perspective of what is meiosis, how do you need to understand it in order to answer any questions about it on your unit test or on the AP exam. Again, please let me know if anything was not clear, and let me know what is your current understanding of meiosis, and do you think it's a level 1, level 2, level 3, or level 4 understanding? Have a great day, everyone.